the CEO of Edgewise. In clinical papers looking at histological samples from Duchenne muscular dystrophy patients, it was postulated that one type of muscle fiber, fast muscle fiber, selectively degenerated, and that regulation of the damage to that muscle could be used as a therapeutic intervention. That led to the founding of Edgewise. Our vision has become the leading muscle platform company, creating precision medicines for the treatment of debilitating and genetically driven rare muscle disorders. This is our disclaimer. Our vision is to improve the lives of patients and families suffering from rare muscle disorders. We regulate key muscle modulators that protect the muscle from injury. Our lead program, 5506, is focused for the treatment of Becker, Duchenne, and other muscular dystrophies, including limb-girdle. We've successfully initiated the phase 1 healthy volunteer studies, and we plan to initiate patient studies in Becker muscular dystrophy this year. I'd like to just introduce myself, Kevin Koch. I was trained in biochemistry, have worked in drug discovery and development for over 30 years. I started my career off at Pfizer and Amgen, working for 10 years in designing drugs for the treatment of inflammation and rheumatic arthritis. I then founded a company called Array Biopharma back in 1998. We were quite successful. I was the president and chief scientific officer there for 15 years. The company put over 25 molecules into clinical development, and we have 6 drugs on the market treating patients. I then worked for Biogen for a couple of years, where I was part of the team that helped develop Spinraza for the treatment of spinal muscular atrophy. And over the past 5 years, I joined OrbiMed, which is the largest healthcare dedicated private and public equity firm, where we invent and put together new companies and invest in companies to treat diseases with high unmet medical need. This is where I met Alan Russell back in 2017, and I'd like to now let Alan introduce himself.
Hi there, this is Alan Russell. I'm a pharmacist by training, originally in the UK, did a PhD over there, and then came to the States to work at Stanford Medical Center as a postdoc before starting my industrial career at Cytokinetics. This introduced me to skeletal muscle physiology, and I've really been developing drugs in that area for almost 20 years now. So I spent 9 years at Cytokinetics and then was at GlaxoSmithKline as the head of their muscle metabolism research unit, interested all the while in skeletal muscle physiology, disorders associated with skeletal muscle, both muscle wasting type disorders and also the myopathies that you're all so familiar with, before approaching OrbiMed back in 2017 with this self-contained idea that we'll tell you about today.
So we put together a very experienced and seasoned executive team to invent and develop our drugs. Mike Carruthers, our chief financial officer, worked with me at Array, also worked to bring Novalis up public, which was a rare disease company working in the CF space. Bered Durakshan, our chief business officer, who spent the last 15 years developing drugs for the treatment of multiple different rare diseases, including diseases in pediatrics. John Moore is an expert in the contractual arrangements that drive forward our clinical trials and make that a reality. And then Abby Bronson, who many of you know, was at PPMD, a key asset to our culture and our company, in that she has many interactions with our patients, the KOLs, and the FDA. I think we value this type of capability because in the end we're doing this for the patients, and it's really important to connect with the patients and make sure we are doing things that really address their needs. We have a fantastic group of advisors, many of them well known in the field. Leslie Lanwan was one of the founders of a muscle company, cardiac muscle company, Myocardia, She's world-renowned in the molecular biology of muscle. We reside at the University of Colorado BioFrontier Center, where we have our labs and our offices. Lee Sweeney is world-renowned in the preclinical models of Duchenne and Becker muscular dystrophy. Barry Byrne and Craig McDonald, very well-known clinicians who have developed many drugs and run many trials in the neuromuscular space in Becker, Duchenne muscular dystrophy, as well as Pompe disease. And then John Vissing, well-known in the field of adult neurology, one of the premier trialists developing drugs in Europe. And of course, we have intense interactions with all the different patient advocacy groups. I'd like to thank the MDA for providing this opportunity for us today to provide some information to you about our drugs. And I'll turn it over to Alan.
Okay, so I'm going to spend the next five minutes or so giving you some background on skeletal muscle, and the special role that dystrophin plays in skeletal muscle, and then a little bit about the strategy that we're employing to try and stabilize these muscles. It's really a very high-level presentation. I'm not going to go too much into the weeds, but hopefully you'll leave this presentation with a little bit of an idea about what we're trying to do and the time frame that we're trying to do it on. So what you're looking at here is a graphic of muscle attached to bone. So we have graphically sliced right down the middle of that muscle. And what you can see is all the little muscle fibers there lying in parallel in that tissue. And what we've done is we've blown out one of those muscle fibers just to show you where the dystrophin complex is and why it matters so much. So dystrophin is often described as a shock absorber. I would like to maybe posit it slightly differently, that what it's doing is connecting the contractile stuff in your muscle to all the outside material. So there in the complex there on the bottom, what you're looking at is this rather complicated organelle that spans between the basement membrane and the contractile machinery. And the basement membrane is just a fancy way of saying the connective tissue that surrounds all of our muscle fibers to give them support. So what you can see there is a complicated structure that has really one primary goal, and that is when the muscle contracts, the membrane that it's attached to contracts also, as does the basement membrane, so that so that everything moves in a unity. It's a very complicated organ, and it needs all of this structural machinery to hold it together. So you can imagine if you don't have elements of that complex, for instance, if you have a mutated or an absent dystrophin, or if you have mutations in the sarcoglycans, for instance, or indeed in the laminins on the outside of the muscle, when the muscle contracts, you get this discordance between the membrane and the extracellular matrix, where that is not contracting to the same extent as the internal machinery, and that really is what causes the injury. And we'll show you that in action, actually, in one of these videos that we have later on. Now, I'm just going to delve briefly into the complexity of skeletal muscle, hopefully without confusing things too much. So I think many of us think of our skeletal muscles as just a big block of the same organ, but we're actually made of different types of fibers. We're made of slow and fast skeletal muscle fibers, and the body has cleverly designed this machine so that we can do lots of different things without getting tired and maintaining our performance. The easiest way of thinking about fast and slow muscle is dark and light meat on a chicken in many ways. So dark fibers, they tend to be slow. They contract more frequently and they fatigue slower. So they are really your workhorse fibers. Fast fibers, those are the light meat on the chicken. They are faster contracting, they are higher power generating, so they move faster and thus generate more power, but they fatigue quite fast. So for instance, in power athletes, they're really honed for fast fiber activity, whereas long distance runners are often honed for slow fiber activity. It so turns out that fast fibers are more susceptible to injury in a variety of inherited muscle diseases, including Becker and Duchenne, but we believe probably extending into the limb-girdle muscular dystrophies too. So there were papers in the late 80s and subsequently describing how fast skeletal muscle fibers appeared to be getting injured more rapidly than slow ones. And we've just added to that literature actually with a publication that we published in Muscle and Nerve Journal showing biomarkers of this observation too. Now, we are made of approximately 50% fast fibers and 50% slow fibers, and they're spread in a mosaic throughout our muscles in order to get us where we need to go. Our strategy is specifically protecting these fast fibers in your skeletal muscle. So there's a lot of information on this slide. I'll just run through it very simply for you. So as I described, the fast fibers appear to be more susceptible to injury in your body. It's not truly known exactly why that is, but it's probably associated with the speed of contraction and how much power those muscles generate. We're going to try and selectively protect those with our medicine 5506. So we've made this compound 5506 that's designed to protect your muscles from injury. Okay, it's a highly selective muscle modulator that actually goes into your skeletal muscles and selectively protects those fibers. In preclinical models that we've run in dogs and mice, and we'll show you some dog data actually, what we found is that this stabilizes the muscle, protects the muscle from membrane injury and the kind of leak associated with you observing CK in circulation, for instance, and long-term helps to protect you against fibrosis and functional declines associated with the prolonged inflammation and degradation of the muscle. So, by working at the level of muscle to stabilize it and protect the membrane, we believe we're right at the top of the disease cascade. If you think of dystrophin absence disorders such as Becker and Duchenne as really connected between when the muscle contracts, the membrane becomes injured, and the muscle degrades, we're right at the top of that cascade. What we're hoping to do is stabilize the muscle membrane, and thus prevent all of the downstream things that happen as a result of that stress, such as the breakdown of the muscle itself, the inflammation associated with that breakdown, the need for the muscle to regenerate after it breaks down, and then the repeated cycles of this that cause your stem cells in the muscle to degrade and eventually lead to replacement of muscle with fibrotic or fatty tissue. Now, this is a strategy that's not been tried before. We always use the big word orthogonal, which is a weird word, but it means it's just coming in from a different direction. We're not trying to change the gene status of muscle. So it doesn't really matter for our therapy what form of mutation that you have. The medicine should be appropriate for both Duchenne and Becker, but also, as I mentioned, for the limb-girdle muscle myopathies as well. And because it's coming in from a new angle, we should be combinable with genetic approaches such as antisense oligos or gene therapies that are in development, or in terms of the downstream type of interventions such as the glucocorticoids. All right, so this is the compound in action. And one thing I really like about this is not many people are used to seeing dystrophic muscle actually breaking down. And that's exactly what you're looking at here. So these muscles are isolated from a mouse with muscular dystrophy. So this is the Duchenne model called the MDX mouse. So this muscle has been isolated and it's contracting in a little bath. And the left-hand side muscle has not been treated with the compound, just with a little bit of vehicle. And on the right-hand side, we put a low concentration of the compound, 5506, into the media around the muscle, and it's absorbed into the muscle in about an hour. So this is 1 hour of pre-treatment. On the left-hand side now, what you're observing is that membrane stress. So the muscle is contracting, as muscles do, and then what's happening is because you have this discordance between the membrane and the contractile machinery, that stress causes these degenerative events in the muscle. You get calcium coming into the muscle, and it turns on all of these pathways that cause the fibers to become chopped up and degrade. And that's what you're seeing here as these balls appear to be retracting along an axis. It's actually the whole fiber collapsing. Now, as you can imagine, as that kind of collapse occurs, what the body needs to do is dispose of that material and then grow a whole new fiber back by mobilizing the little stem cells that exist around the outside of the muscle fiber. On the right-hand side, what you're seeing is the same type of muscle. So this is a dystrophic muscle, same as on the left, but pre-treated with the compound. It's still contracting, but what we've done is stabilized internally the myofilament. So the compound is bound inside the muscle, and it's changed the relationship between that contraction and how much the membrane is getting stressed. So we don't see those types of retractions. We don't see the accumulation of these balls of muscle. The fiber is maintaining its function as if it was a normal muscle, but of course we haven't replaced the dystrophin. So of course, this is a preclinical model. You're looking at a mouse muscle in the dish. That's quite a long way away from a muscle in the leg or an arm of a patient. But if we could achieve this in people, of course, there would be a profound impact upon long-term health of patients. Here's the one piece of preclinical data that I'll show you, and we're quite proud of these studies because they are very challenging to do. So what we did is we did short-term dosing of this compound 5506 in dogs with muscular dystrophy. Much the same as people, dogs exhibit muscular dystrophy. In this case, this is a golden retriever that has muscular dystrophy. And some colleagues that we work with at Texas A&M have a colony of these golden retrievers, and we use 4 of them for these studies. Now, what we did is we waited for the disease to manifest in these dogs. They tend to be born fairly normally, and then they progressively weaken to about 6 months of age, and then their function stabilizes. So this is kind of like a disease-stable dystrophy model. The estimations are it's kind of akin to a 10-year plus Duchenne patient. They have no dystrophin. What we did is a 2-week study and looked at biomarkers on the left-hand side. So as you probably know, in these types of diseases, the muscle becomes aggravated and leaks proteins into circulation. And one of those famous proteins is creatine kinase. It's often the first way that you diagnose muscle myopathies. And what we observed is pretty much from the first dose of the compound, we saw approximately a 60% reduction in CK, but then reversed when we took the compound away. You're just looking at the compound effect there on the left. So when we gave the compound, CK went down. Now, in a separate set of studies, we actually asked whether the dogs were more or less active. Of course, we could decrease CK by making the dogs less active. But what we actually found was the opposite. We fitted the dogs with activity monitors that you can buy for your pet, called FitBarks. This is a really handy device that you put on your dog, and you can actually track their activity with your phone. And what we noticed when we gave the compound is we saw a 30% increase in physical activity during the dosing period. So not only were the biomarkers going down, activity was going up, suggesting that we've changed the overall health of skeletal muscle. Okay, I'll hand it back to Kevin now, just to give you an idea of where we are on the clinical development.
Okay, thanks, Alan. So we've initiated our clinical trials. We've completed our single ascending dose studies. So we keep increasing the dose every 2 or 3 weeks in healthy volunteers. And we look at safety and tolerability. And what we found was the molecule was generally safe. And we had it reviewed by a independent data monitoring committee, and we are now moving into a multiple ascending dose study where we'll dose the normal healthy volunteers for 14 days. That will be starting in about a week or so. After that, we will move into phase 1B and 2 studies, where now we test the drug for the benefit and safety in muscular dystrophy patient populations, which would include Becker first, but then Duchenne a little bit later in the year. Finally, what we'll end up doing is when we have seen the appropriate amount of information. We understand the safety, how much to dose, how it would affect people's function, how it might affect the damage to the muscle, and what exposure and dose would give that optimal result. We'll then go into phase 3 studies. We'll run a controlled study for approval with a placebo arm and perhaps 2 doses of the drug, and each individual disease will have its own phase 3 clinical trial. So tell you a little bit about the product candidate. It's a pill. It's quite small, probably on the order of 10 or 20 milligrams, which is about the size of a Tic Tac, or it can be dosed in a liquid suspension, or it can be chewed. So it's a very straightforward drug to deliver. It's designed to protect the susceptible muscle fibers in a highly selective way. In studies in mice, we've shown protection of both the skeletal muscles, but interestingly, the cardiac muscle as well, which is really important in these disease populations. It's potentially appropriate for any age. It does not depend on the mutation status of the patient or what background therapies you're on, and it's applicable for all types of muscular dystrophies. So thank you so much for coming to our presentation and learning more about Edgewise. There's information. We have a website, and you can go to info@edgewisetx.com. And there are many detailed discussions of the science and the medicine on an SEC website regarding our company and our plans. Thank you so much.
I'm just looking at the comments now, and we've had a number of really good questions. So thank you very much for the engagement. The first one is just about recording. Yes, this will be available for you to check out later, and there'll be a link shown. So not a problem there. Would this medicine, this is from Hannah Lowe. Thank you so much, Hannah. I believe this medicine would benefit kids with congenital muscular dystrophies, so non-dystrophin related. I do believe that there's a large family of disorders that may be appropriate for the compound. The congenital ones can be complicated. Certainly our priority is Becker and Duchenne with some limb girdles, but happy to engage on the possibility of congenital muscle myopathies. From Kyle McMahon, is there an age limit to where this therapy would be beneficial? Hypothetically, no. If you have muscle left, there's the potential for protection of further degradation. Now, we do think that the dose response for the compound will be different as you get older. We'll be starting studies in milder disease, but as we go forward and understand more about the compound, we will be interested in exploring more severe disease. So that will be tackled in good time. We have from Kathy, will 5506 increase strength or rebuild muscle in those who've already become weak from muscle wasting? Now that's a great question. Of course, once you've got fibrosis and fat replacing tissue, it's very difficult to replace that lost muscle. And that's the same for any intervention, unless you could somehow graft new muscle in. Now, saying that, the dog data that we showed you earlier did suggest that you can acutely improve function with the muscle that you have left. And our hope is by changing the relationship between how muscle contracts and how much it degrades, that the muscle that you do have remaining can be more healthy and thus more functional. You know, I think one of the challenges that myopathy patients face is, of course, that we understand that the more we use muscle, the healthier it becomes. But of course, that relationship is changed in these patients. So if we could change that somewhat closer to a normal patient, you would indeed hopefully see some improvements in physical function. From Roberto, are there any effects related to prednisolone use? Our orthogonal, our different strategy is not related to prednisolone, no. You know, it's been the source of many, many decades of research actually as to why glucocorticoids work. There's some new papers in this area that are very interesting, but it is a complicated story about why those kind of compounds work. Our compound is directly binding into the muscle to stabilize it, and as such is quite different from prednisolone.
So we should not have the safety issues of prednisone, for sure.
How can one volunteer to participate in trials? This might be a good one for Abby to answer if she's there. Maybe she's gone.
Yes, I'm here. Hi, everybody. So glad that we're at the stage that we can talk to you all. So yeah, so there's been a lot of questions on the chat and also the Q&A about how you volunteer for these trials. Are they only in Texas? Now, the email address that we had up at the end, info@edgewisetx.com, eventually gets to me, and I reach out to every single one of you individually. And if you want to set up a call, I can do that as well. So email and to that email address, and I promise I will answer you.
So in regarding the trials, though, to be more specific, there is the trial that we have on clinicaltrials.gov for 14 days in Texas, but we are planning additional trials, which will be more than 14 days, which will start probably in the fall. Initially for Becker, but as soon as we can get into Duchenne patients, we'll get into Duchenne patients as well.
So we do have a question from Mario around that topic about whether we've selected all our candidates, and the answer is absolutely no. Your participation is highly encouraged. And from Doug, will this reverse weakness in, say, a 30-year-old DMD patient, or is it prevention of deterioration? I kind of touched on that one a little before. You know, once you've lost muscle, unfortunately, it's difficult to replace that muscle, but what we can hopefully do is improve the health of the muscle that is remaining. How do you sign up to be a patient for the trial? From Anthony, thanks for the question. That I think hopefully Abby addressed that. I think that might do it. Did I miss any questions, NDA?
One just came in on the chat. It says, what would be the length of these trials and would there be placebo?
I'll let Kevin answer that one.
You know, so initially what we'll do is we'll have studies where we will not have a placebo, and we'll look to try and identify the appropriate dose, and we'll monitor the patient closely over time. The initial trial in worldwide, as I said, was 14 days, but beyond that, we plan to be in a position where we can dose patients indefinitely on the drug, starting in about September. So with that in mind, people who come onto that study could get drug for as long as we're developing the drug, which could be years. So yeah, I think there's a huge impetus to join these trials because I think it could really benefit you.
We have had another question. Would 5506 stop you from taking future treatments if other treatments come out? This is from Kathy. Kevin, you want to go on that one?
No, no, actually, you know, because it's a small molecule, it's an oral pill that leaves your body over time. Once it left your body, you should be able to go on other trials. So that's the great thing about this type of medicine, because you can be washed out, so to speak, and then you can start on something new.
And I have a follow-up for you from Mario. Is there a projected timeline to complete trials and get to the public?
So we think we certainly have a vision. We would go as fast as we can. There's various ways in which we work with the FDA. Sometimes it can be an accelerated approval, meaning that with a very small number of patients, you can get to the public very quickly. And it depends if the magnitude of the clinical benefit we see is very high. Sometimes the agency grants that very early proof and allows you to go sell the drug and provide it to the public. Otherwise, a more standard approach is to run usually a multi-year phase 3 study, which can be longer. So it really depends on how promising the medicine is and the results we see over the next year or two. But realistically, if you participate in the trials, you can have the drug for as long as we're developing the drug.
From Brandon, what feedback have you gotten from the primary doctors of muscular dystrophy patients? You know, we have been kind of habituating these conversations for the last couple of years, you know, visiting a variety of different doctors around the country. I mean, this is really just the start of that journey, though. I would say most physicians, much the same as you guys will, you know, as you're adopting this, they take a while to go, oh, there is a different strategy that you could try. And then I think what they really like, A, it's a pill, B, it's appropriate for a broad population of patients, and C, it's potentially combinable. So we've had pretty good feedback. Would you have anything to add on that, Kevin?
Yeah, I would say that we've had multiple, now this is the fourth interaction with patients that we've had, and there's been a lot of enthusiasm from the patients because it's so new, and it's essentially it's a biophysical approach. You're affecting the physics of the muscle, and by doing that, we're quite clear that we will see an effect, and I think it will be a highly beneficial effect. And I think this whole concept of protecting your muscle is a key aspect to the disease. If the muscle's there, if you can save the muscle, that will create a better option for patients.
We have a couple more. So from Charles, I believe, Charles, an ICD would be an implanted defibrillator. Does this disqualify me from the trial? Potentially from the early studies. We are going to go into healthier patients in the earlier studies, but of course in the later studies that we hope to roll out afterwards, we would have broader inclusion criteria that include patients with underlying cardiac disorders. Let's see now. Would gene therapy patients be excluded from any DMD studies? Kevin?
No, I think, you know, as these the gene therapy studies can be worked. I mean, so we could be treated first and then dose the gene therapy. We haven't discussed this with any of the companies with gene therapy. They could be dosed together. And if you know, depending on the results of the gene therapy, perhaps adding our drug after you've had gene therapy would be the appropriate approach. So this could be used in any one of those cases.
We have, gosh, great questions. What possible side effects have emerged in the canine and healthy volunteer trials? Kevin, you want to summarize that?
Yeah, so what's interesting about, because we're selectively targeting a particular muscle, what we see is essentially in the rodents and the preclinical and the dogs, is that wherever the fast muscle is, you sometimes will see some effects on that muscle. In humans, because we're a mosaic of a mix of fast and slow, we don't see the same types of side effects that we see in the preclinical models. So what we've gone by is there are a set of patients that are missing the fast fiber. So there are patients who have a mutation, so they have no fast fiber, they're entirely slow fiber. And those patients are ambulatory throughout life. They have some facial and some neck associated weakness, but they are generally normal and they have normal lifespans. So what we've seen preclinically, or what we've seen clinically, is the first thing we see, which is quite an interesting result, is sleepiness, which is quite benign. It's readily monitorable. It's associated with the first 2 or 3 or 4 hours of the dose, and then after that it goes away. And we think that we can dose much lower. So how you run a clinical trial is you dose as high as you can go, and then you see what you see in the patient, and then you dose where you think the drug will be effective. And so we think the drug will be effective at a much lower dose. And what you saw at the high doses was something that was relatively benign. So we're quite pleased with the result of that. And we look forward to additional studies when we dose it multiple times or start dosing in patients.
We have a couple of roundup questions, and then I think we're going to draw the event to a close. So thank you so much for your questions. Just a couple of roundup ones. We had one about the location in Texas and whether other sites would be available. So for this first study, it's going to be Texas only, just for the 2-week dosing. But for the longer term studies that we'll be doing afterwards, those will be multi-center across the country and possibly also in Europe. So a lot of options there for you.
Yeah, and for the longer studies, we will transport you and pay for all your travel if that's required. So we will make it as easy as you can to participate.
Has the COVID vaccine testing had a positive impact on your testing from a time to market perspective? We had some early delays, but things are clocking on okay. I mean, fortuitously for us, the delay between finishing our animal studies and starting our clinical studies has largely occurred during the worst of the COVID period. So hopefully we won't get too many negative impacts. Will 5506 help other organs like the heart? Possibly. In our preclinical studies, we did see positive effects in the heart in a really severe muscular dystrophy model. So we really hope to see that your whole body skeletal muscle health knocks onto your heart. But of course, people are very different from preclinical species, and we'll only find out with testing. Can we tell you about other programs in your pipeline? Maybe another time, I think. That's a long one. So thank you so much.
Thank you all. It's been a great conversation. I really enjoyed your questions. We look forward to meeting you in person someday.